Welcome to the second session of Biology Made Simple with Samayye. In today's session, we are going to discuss these key points. So, let us first begin with the types of cell division. Now, cell division is basically of three different types. The first one that we are going to discuss is the most common one, which occurs in the body cells or the somatic cells and known as mitosis. Now, as I said, body cells or somatic cells. So, which cells exactly? Let's check few examples. The cells can be that of the embryo. It can be of the plant meristem, which is again the region of actively dividing cells. Then certain cells like skin cells, blood cells, liver cell, etc. Moving ahead, the next type of cell division that we are going to discuss is the meiosis. And meiosis occurs mainly in the reproductive cells of the reproductive organs. So these cells are referred to as the germ cells that produces gametes. Now, in case of human male, the reproductive organ is the testes that produces the gamete sperm by the process of meiosis. In case of human female, the reproductive organ is the ovary that produces the gamete, that is egg cell or the ova. So the germ cells present in the respective reproductive organs will give rise to gametes. The third type of cell division present in lower organisms is the amitosis. Now, amitosis occurs in organisms like unicellular yeast, amoeba, and meganucleus of Paramecia. Let's move ahead by understanding the difference between mitosis and meiosis. First of all, as I already said, mitosis occurs in the body cells or the somatic cells, whereas meiosis occurs in the reproductive cell. Now, what is the significance of mitosis? How does mitosis helps? Let us check. The first thing is for the purpose of growth. Next, for healing or repair of tissues. Say for example, there is bruise on your hand or your leg. After few days, you will find that your wound is completely healed. This is all because of the cell division, that is mitosis, which helps in healing of wound or repairing of tissue. Mitosis is also significant during development of the embryo. So right from a single celled zygote to a multicellular organism, here I have checked the embryo of human being. So you can see over here, right from the fertilized egg, there is a 2 cell stage, then there is 4 cell stage, 8 cell stage, and so on. The cell divides by the process of mitosis, giving rise to this multicellular fetus. Okay, so helps in growth, repair, and development. Meiosis, as I already mentioned, it helps in the formation of gametes. That is, in case of human male, it is sperm. In case of human female, it is the egg. So it helps in formation of haploid gamete. Next, in case of mitosis, the newly formed daughter cells are diploid. That is, when a parent cell divides, it gives rise to two daughter cells, and both the cells are diploid, containing same number of chromosome like that of the parent cell. Unlike in meiosis. Now what happens in meiosis is it gives rise to 4 cells which are haploid. Whatever is the number of chromosome present in the parent cell, half the number of chromosome will be present in the newly formed daughter cell. That's why we are here using the term as haploid. Half the number of chromosome, hence haploid. Now in case of mitosis, as the daughter cell carries same number of chromosome, equal number of chromosome like that of the parent cell, it is also referred to as the equational division. Whereas in case of meiosis, the newly formed daughter cell will have only half the number of chromosome. The chromosome number reduces to half, hence it is known as the reductional division. Now, mitosis division is going to occur only once. Meiosis, it is going to occur two times, twice. That is, the first stage will be known as meiosis 1 and the second stage will be known as the meiosis 2. Now, here you can check in mitosis, there will be formation of two daughter cells and both the daughter cells are diploid, carrying same number of chromosome like that of the parent cell. But in case of meiosis, the parent cell is diploid, but after division, you can see there are four newly formed daughter cell which are haploid. Clear with this part? Okay. Yes. Now next session is regarding the next topic is regarding the cell cycle. From the name itself, term itself, you can come to know cycle means something that is repetition of events. Now here cell divides at regular intervals and that's why it is referred to as the cell cycle. Now if you check this particular diagram, you will come to know. Okay, before that, let's check one example. If your mama wants to prepare burger for you, she needs to do certain pre-preparation like making of burger buns or burger patty and also whatever the essential sauces that are required or the veggies. She needs to take care that all of this is ready before making the or before combining the actual burger. Same happens in the cell. Now when cell decides that it has to divide, so first of all it needs to prepare itself, and that's why you will find there are two main stages in cell division. The first one is the preparatory stage, you can say, which is known as the interphase. So here you can check G1, S and G2. These are the phases or stages of interphase, you can say, wherein there is synthesis of DNA, synthesis of other cell organelles that are required. And in case if there is some problem with this cell, then these are the checkpoints. So in this checkpoints you will find that care has been taken so that if there is some problem with the cell itself, the cell will not divide. Okay, and if everything is occurring smoothly, then cell will enter the second phase, which is known as the mitotic phase or the M phase. So here you can check. Now mitotic phase has two parts. Once again, the first part is known as the karyokinesis or the nuclear division. Now this karyokinesis or nuclear division is again subdivided into four different stages. To begin with is the prophase, metaphase, anaphase and telophase. So once division of nucleus is completed, the next part is the division of cytoplasm, which is known as the cytokinesis. So this is everything about the cell cycle, the events that are occurring in this cell cycle. Now, the between two cell division, the time interval that is there is known as the generation time, and cell cycle duration is different for different cells. Here you can just check. In bacteria, it's just 20 minutes. Onion root tip cells will take as long as 20 hours. Humans again 24 hours. Yeast cell it will be 2 to 3 hours. Okay, let's check on to the mitosis. Now in this session we are going to focus on mitosis. Meiosis we are going to see it in our next session. So this session is all about mitosis. Here you can see. Now this mitosis term was coined by Walter Fleming. As we already discussed, there are two phases of mitosis. Now you are quite aware of which are these two phases. The two phases are, yes, karyokinesis and cytokinesis. Karyo, division of nucleus, cyto, division of cytoplasm. Now let us talk something regarding the cytokinesis. Cytokinesis in case of plant cell and in case of animal cell there is some difference. So you can just look over here. In case of animal cell you will find there is formation of a cleavage furrow, and in case of plant cell you'll find there is formation of a cell plate. So just look over here, cell plate formation, and the two cells are getting separated. So animals it is by cleavage, and in case of plant cell it is by cell plate formation. So these terms we are going to need while studying more about the mitosis. That's why I have shown this part. So here you can see this is the unreplicated chromosome. The two arms of chromosome are joined by the centromere. So here centromere is there. After replication, the chromosome shape is somewhat of this kind, and these two are referred to as the sister chromatids. Okay, now if you consider one chromatid from this blue part and one chromatid from this red part, you can call it as the non-sister chromatid. So you need to understand the difference between chromosome. Now this entire part is known as the replicated chromosome, and this individual part is known as the chromatid. Next, you need to know what is centriole. So this organelle is known as the centriole, and these are the spindle fibers, the special type of proteins that help in mitosis. The central plane is known as the equatorial plane. Also, why the names stages of karyokinesis are named as prophase, metaphase, anaphase and telophase. Pro for first, meta middle, ana up, telo end. So, ready for stages of mitosis now? Okay, the first, as I said. So, it has to be obviously prophase. So, what happens in case of prophase is condensation of chromosome is going to start. You'll find initially the chromosomes are in the form of a very thin thread. So, here condensation will start and the chromosome will start to become short and thick. They will start to pair with the sister chromatids. So, here you can see it clearly, pairing with the sister chromatids. Also, the centriole will duplicate and each centriole will start moving towards the opposite pole. Also, the nuclear membrane, it will start to disappear, the outer covering of the nucleus, and also the nucleolus, it will start to disappear in prophase. Prophase is followed by the second phase, that is the metaphase. So here you can check the diagram of metaphase, wherein there is no nuclear membrane. Condensation of chromosome is also completed. Chromatids are paired and they line along the equatorial plane. And there is formation of spindle fibers, a special type of protein that is also completed, and it initiates the third phase, that is the anaphase. So here you will find that the centromere it splits. Okay, it splits, and that's why the chromatids they get pulled apart towards the each of the poles. So when the chromatids reach at this particular juncture, it appears like bunch of bananas. Now here you can imagine what will happen if these chromatids are pulled towards each of the pole. It appears like bunch of bananas. And then there is our last phase, that is the telophase, wherein the chromosomes at opposite poles will start to decondense. They will once again start to become thin and invisible. Nuclear membrane will start to appear, and hence there is formation of two daughter nuclei. The cytoplasm is still one, but the nucleus are two, so two daughter nuclei, and this will initiate the next phase, that is cytokinesis. In telophase there was division of nucleus. In cytokinesis there is division of the cytoplasm. Concentrate on this particular yellow colored part. So this completes the mitosis cell division. So these are a few questions and a summary of mitosis. Okay. Thank you.